Okay, sports fans, welcome back from a wonderful Christmas break. We are going to kick butt with a little photosynthesis today. Now, up until now, we've looked at sort of the overview of it, what's made, why it's made, and so forth, and I'll review that a little bit. But today, my friends, we are going to get into the serious nitty-gritty of this. This is going to be a challenging video, I think. Work hard on it. You may need to watch it more than once. If you don't understand it, write down lots of questions and we'll discuss it in class. But really, at heart, this is just a step-by-step process. And if you understand what goes on at the end of this, I think you're going to feel powerful. You're going to feel like this is so cool that you understand what's going on. So, here we go. All right. Really, what we're focusing on today is the light-dependent reaction. In other words, stage 1 of photosynthesis. Okay, here's some things I want to review. Here's the point of photosynthesis. The point is to convert radiant energy or sunlight into chemical energy or sugar, specifically glucose. There are other sugars that are made potentially, but glucose is the one we focus on. Okay, that's the point of photosynthesis. Remember that. Now, second, what does photosynthesis? Well, here's some organisms. All the organisms that do photosynthesis are called producers because they produce the energy that all the other organisms on the planet need. Okay? Producers, of course, are plants. We're very familiar with that. And algae, which algae are a type of plant, but they're kind of their own specialized type of plant. So plants, algae, and some protists, which are single-celled organisms. Okay? If they do this, and to do it they need chloroplast. We'll talk about that in a second. Now, this is where we start getting more interesting. There are two stages to photosynthesis. There's the light-dependent reaction, which is what we're going to talk about today, and there's also the light-independent reaction, which is called the Calvin cycle, after Melvin Calvin. And he's the one that discovered the cycle and figured out how it all worked. Okay? Some people call it the dark reaction. That's pretty much out of fashion now because the light-independent reaction does not require light, but it can certainly happen in the light. So they don't usually say dark, they usually say light-independent. Doesn't need it, doesn't matter if it's there. Okay, but again, today we're going to look at the light-independent. Now, the first thing I want you to realize is what are we using to what's doing all this work? And it's the chloroplast. Okay? So let's look at the chloroplast. This, of course, is inside a plant cell. There's lots of them inside a plant cell. And if you look at a leaf cell, you might have 50 of these chloroplasts inside one single leaf cell. Now let's look at how it's made. It has two membranes around it. And these membranes are the phospholipid bilayers, just like you've seen before. Okay? There two of these phospholipid bilayers, one, then another phospholipid bilayer. Now, inside that is this liquid, this jelly, and the liquid is called stroma. Okay? And we're going to look at that closer in just a minute, but the jelly liquid inside is called stroma. These discs here that look like kind of green coins, maybe leprechaun coins, right? They are called thylakoids. And right there is how you spell it, thylakoid. Now, a stack of these thylakoids is called grana, and granum is more than one, is a plural of it. So grana or granum is a stack of thylakoids. And then connecting these grana are things called stroma lamellae. Stroma lamellae. It's just fun to say. Okay, that is the basic structure of this. But what we're really going to focus on today, because we've done this before, what we're going to focus on today is one individual thylakoid. Okay, we're going to focus on that. So, let's look at that. Okay, so here's our blown up version. This, my friends, is a single thylakoid. What you see right here in this circle. This little single disc, that's this. Okay? And it has lots and lots of parts to it. So let me show you what those parts are. The first part is, of course, the thylakoid itself. But inside the thylakoid is a fluid called lumen. Okay? That's what this light green is this jelly or this liquid called lumen. The black line of the thylakoid is just a membrane. Again, if it's a membrane, it's a phospholipid bilayer. So if I was getting up really close here, then I would see that it's made just like the cell membrane. It's got the phospholipids like this, and there's two of them, and they go back to back, and it's a nonpolar membrane, just like we've done before. Okay? Okay, now in that membrane, we have proteins like we've looked at before, and this little blue looks like a V right here. There's several of them. I think there's 5 of them. That is a protein pump. Okay? It's a protein pump, and we've talked about protein pumps before. So I'm going to put that right there for it. We've talked about protein pumps before for insulin, right? Insulin getting inside of the cell membrane. Well, and I told you at the time that protein pumps require energy to work, okay? And I also told you that they either move great big things like insulin, or like glucose in, I'm sorry, or they move small molecules against a concentration gradient. And that's what's going to happen here. This hydrogen is a very small atom, and it is going to be pushed through the membrane into here to collect a whole bunch of stuff. We'll get to that in just a minute. But that's what this protein pump does. It's specialized for hydrogen ions. Okay, the next thing are these little red dots, and those are called electron carriers. And we'll explain what those do in just a moment. Electron carriers, maybe I need to put some arrows here. So that's that, and that, and that. Okay, protein carriers are going to be kind of a, I don't know, sort of a floating base along the membrane. It's just a protein in the membrane. We'll talk about that in just a minute too. Now these two big guys are really special. The first one is called photosystem 2. And I know it's this gold one right here. And I know you're going, 2, well, where's 1? That's this one. We'll talk about it in a minute. It's only named because of the order they were discovered. It doesn't matter which one's which. It's just they discovered this one after they discovered this one. Okay, so the other one is photosystem 1. And we shorten it to say PS2. You guys should be able to remember that. Or PS1, right? So PS2, PS1. We're going to look at PS2 really close up here in just a second. This big gray guy down here at the end is called ATP synthase. Okay? ATP synthase. And it is really a channel protein. Remember we have aquaporins that let water in and out? Well, this is a channel protein that lets hydrogen ions out, but at the end it's specially adapted. See this little funky thing here? That's the synthase part of this, and it will actually spin. So as a hydrogen ion goes through here later, this thing starts to spin, which is, to me, is just profoundly cool. It's even more cool that they figured it out, but there it is. All right, so that's ATP synthase, and we're going to talk about how all that works in just a minute. Okay, this little yellow guy down here, this little yellow triangle, is what's known as a hydrolysis enzyme. Hydrolysis, what the heck is that? I'll get to it. Just wait for it. Okay, and then finally, outside the thylakoid, whoops, I lost my guy. There it is. Whoops. Outside the thylakoid is the stroma. Remember we call that's that fluid right here, all around here. All that fluid is the stroma, so that's what all this is. Okay, are you confused yet? I know, there's a lot of stuff to know. So you got to know a lot of the parts before you can know any of the steps. And we're going to get to that. So I'm about to tell you all about it. But I want you to kind of take a minute and make sure you have a diagram of this written on your paper, so that as I talk about it, you can actually kind of go along with me with your diagram. Okay, so I'll wait just a second and let me know when you're ready. Are you ready? Are you ready yet? Okay, pause it. Okay, I'm tired of waiting, so just pause it. Okay, now let me reorganize this for a second. Okay, before we go much further, I need to show you this PS2, this photosystem 2, a little closer. And here it is. This is this. Same thing, okay? Inside this is chlorophyll. And we've heard of chlorophyll a whole bunch, okay? So chlorophyll are those little green circles inside this PS2, this photosystem 2. That's going to become really important when we talk about step 1 in just a second, okay? So, I guess it's time for that just a second. Yeah, all right, so let's move this down so you got it. Actually, I'm going to move that away so you got it. Move this down. Now, inside these chlorophyll, they are made of molecules and made of atoms, just like everything else. And atoms is made of protons, neutrons, and electrons. Okay? So that's a really important part, is that electron. That becomes a big deal. So let me show you step one. You're going to have to write this down. Step one, sunlight hits chlorophyll. So here it comes. Light from the sun comes along and hits this. Okay? And that's a lot of energy. Those photons that we talked about, they contain a lot of energy. And they hit this chlorophyll, and the chlorophyll absorbs that energy. And it's trying to hold on to it. You ever tried to hold on to like a little puppy that's like 3 months old and it's wiggling everywhere and it's so hard to hang on to because it's full of energy? That's what this is like. Well, hopefully you didn't drop the puppy, but in this circumstance, the electron, right there, that little dot, that electron actually jumps off. Ah! Okay? It's so excited, it has so much energy in it now, it jumps off. And it jumps off of this onto this red electron carrier. So let's see if I, oh, boom, it lands right there. Okay? So it jumps from the PS2 to the electron carrier. Okay? That's what happens. Now, what happens is our little electron carrier jumps over here to our protein pump. Okay? So our protein pump now has that electron, and it's jumped from the electron carrier over to there. So let me write that down. It jumps from the chlorophyll onto electron carrier, and then here's kind of a big one. The electron carrier, or the electron jumps off the electron carrier and through the protein pump, okay, and starts to pump hydrogen. There's our hydrogen ion. It pumps this hydrogen ion through it into here. Okay? Now you remember, it's protein pump, active transport. It's going against the concentration gradient, just so you know. And that requires, that's right, energy. Where does the energy come from? It comes from this excited electron that's passing through. But it doesn't stop. The electron jumps over here to this guy. Okay? So he jumps. He's jumped from here to here. Now he's jumped to there. Okay? Now he does it again. Guess what? He jumps, that's right, over to here, and over to here, and over to here. And every time he does that, every time he goes through one of these pumps, a hydrogen ion is pumped inside. Until slowly, a big concentration of hydrogen ions starts to build up here. Okay? So that happens over and over, and that's step 4. It's a repeat of steps 2 and 3. This jumps, 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 all the way down. Now, every jump it makes, it loses energy. Okay? It loses energy with every jump. Pardon me. So eventually, this guy's going to be pretty low on energy, and he lands here at the PS1, photosystem 1. Okay, he lands there. Now here, the same basic thing that happened in PS2 is happening here in PS1. Sunlight hits it. It hits the excited chlorophyll, and that chlorophyll sends the electron. For just a moment, that electron bonds with the chlorophyll. And then it gets excited, and you guessed it, it jumps off again. Goes to the electron carrier, goes to the protein pump, which pumps in hydrogen, pumps over, or jumps over the electron carrier, and jumps over to the protein pump. Okay? These little things right here, this electron carrier, protein pump, electron carrier, protein pump, over here and here, that's called the electron transport chain. Okay? And electron transport chain, let's see where to put it. Oh, let's put it here. No, that's stupid. Let's put it there. This is the electron transport chain, and it's the electron jumping from one to the other, losing energy as it goes, but that energy is used to pump hydrogen ions inside. That's what this is called. And there's 2 of them. There's a big one and a little one. Okay? Electron transport chain. Very cool. Very important to remember. Are you with me? Nod if you're with me. Okay, I'm assuming you're all with me. Okay, I'm going to move this away. I'm going to move this away. Now, something you're probably going to start learning about everything, physics, chemistry, biology, is that things don't just disappear and things don't just appear. They come from somewhere. And in this case, we have our little electron, and he doesn't just disappear. He jumps out. Finally, there's nowhere else to go here, but he jumps off into the stroma, out there into that fluid. Okay? But there's something waiting for him there. And what is it? Well, it just happens to be a hydrogen ion. Now, hydrogen ion is an H+. And what that means is that it's a hydrogen atom that lacks an electron. The plus says there's more positive than negative. And we know that if in a hydrogen atom there's only one proton, that means there's no electrons. So this hydrogen ion really loves this guy and says, circle me, circle me, circle me, circle me, circle me. So he does. And this becomes a complete hydrogen atom at that point. Okay? Well, through this whole process, we get a couple of these guys doing this. So we end up making 2 hydrogen atoms. Okay? We make 2 hydrogen atoms here. Now you might be going, big flip. Well, wait for it. Here's what happens. Let's move this out of the way so I can give you kind of the chemical formula. Okay? The 2 hydrogen ions, they combine with, let's see if I can put it there, 4 electrons. I'm sorry, the 2 hydrogen ions combine with 4 electrons. This makes 2 hydrogen atoms. They combine with a funky little molecule, I don't know if I can fit it in the space, I'll put it down low, called NADP+. What the heck? You do not need to know all the words. I'm not even sure I know all the words. Here's the important part. NADP+ means that it also has a plus charge. And it means that it is looking for some electrons to share with. And so these hydrogens, when these, see how there's 4 electrons here? I bet I'm going to confuse you guys like crazy, but bear with me. See how there's 4 electrons right there, and there's 2 hydrogen? To make this balanced, to make this in Happyville, we only need 2 electrons. But we've got extra electrons. Well, they end up circling the hydrogen too, which for a short time makes the hydrogen an H- ion, a negative ion. Okay? But this negative ion is attracted to this positive molecule. The negative and the positive like each other, and so they bond together. And what they end up making is a molecule called, there's the yields, right? Everybody remembers that? NADPH. NADPH is a big player in the Calvin cycle. We have to have it. This is where it's made. This is a big point of this. There's one other thing I got to tell you about happening here in photosynthesis, and I'm going to in a second, but one of the first things you got to remember is one of the big thing that's made in stage 1 is NADPH. Okay? Remember that. Now let me clean this up. Get this written down because this is all about to go away. Okay. Okay, here's step 6, which is what I talked about here with the electron absorbing more energy from the PS1. Okay. Here's step 7, which is me telling you about the electron jumping off the membrane into the stroma, and then this reaction occurring right here, okay, with the NADP being turned into NADPH, and the description of that is right there, okay? So push pause, write these down, because yes, you need to know all these steps, okay? And then I'm going to come back and tell you step 9. And so we have 2 more little things to learn here. I know it's a long video. Bear with me. Pause. Okay, let's keep going. Tired yet? Yeah, me too. But bear with me. I know it's tough, but you guys are troopers. Keep it up. All right. Now, step 9. I'm going to put it up here. Now, if you want to pause and write that down and then listen to what I say, that's great. Or listen to what I say, then pause and write it down. It's fine. They kind of say the same thing. So here's what happens. Basically, now we've dealt with NADPH. Now we're going to deal with this whole hydrogen issue, because the other big thing that's made here is a little bit of ATP, which is used in the Calvin cycle. And here's how it happens. All this hydrogen that got pumped through all these protein pumps has built up here. There's a big concentration gradient now. There's very little out here. There's a lot here. And what starts to happen is it starts to go through this special protein tube right here, And when it hits here, it starts to spin. And it eventually goes through. But while it spins there, ATP is made. And what happens is the energy from that kinetic energy from the spinning is used to bind a phosphate molecule. I'm sure you guys remember phosphate molecules. A phosphate molecule with an ADP, adenosine diphosphate molecule. ADP and phosphate together make ATP. And that ATP is used in the Calvin cycle. And so every time this goes through, boom, ATP. ATP. You know, it keeps happening. And then the ATP that's made is used in the Calvin cycle. Does that make sense? So really the two things that are made in light-dependent reaction, the point of light-dependent reaction is to make NADPH and to make ATP for the Calvin cycle. That's the point. You should write that down. I'll say it again. The point of the light-dependent reaction is to make NADPH and to make ATP for the Calvin cycle. Got it? Good. Now there should be one more question in your mind. And if you haven't gotten this, pause it now because I'm going to take it away. There should be one more question in your mind, and that question should be, if all of these chlorophylls, the electrons are jumping off the chlorophyll, don't they run out of electrons at some point? And the answer is yes, they do. And if they run out of electrons, then no longer are there electrons to jump down this, and no longer can they pump hydrogen in here, and no longer does this reaction happen. Everything grinds to a halt. No more photosynthesis. So there needs to be a way to get more electrons put into these chlorophyll. And here's how it happens. This is where water comes in. It's so cool. The process is called hydrolysis. And the same thing as before. Pause it, write it, and listen, or listen, pause it, and write it. I don't care which, but this explains it. This little yellow triangle here is a hydrolysis enzyme. And what it does is it takes water and it splits water into two. Really into three. It splits it into hydrogen ions, or hydrogen atoms, I'm sorry, and oxygen atom. Now, here's what happens. The hydrogen ions, then electron from the hydrogen atom is peeled off and goes to the chlorophyll. How cool is that? So the water actually or the hydrogen atom, which is right here, let's draw it, okay, the hydrogen atom's here. The electron is taken from the hydrogen atom that was circling and goes over to the chlorophyll to replenish the one it lost. Meanwhile, this is now a hydrogen ion, which all this is kind of happening inside here. It goes and joins this bunch. So that builds up the concentration more. So what's left? Oxygen's left, right? Yeah, oxygen's left. And so the oxygen is released into the air. So the oxygen's left. It exits the stroma, eventually exits the whole chloroplast, the whole leaf, and goes in the air, and you and I breathe it. And so the oxygen we breathe comes from that water being split during hydrolysis. The hydrogen that's there becomes part of this concentration, and the electron that's stripped from the hydrogen goes over here to join back to the chlorophyll, which allows this whole process to keep going nonstop. How cool is that? That also explains to you why water is important and why when there's no water, there's no photosynthesis. Might be a test question. Okay, I know that is a huge amount of information for you. I hope you get it. I think it's totally cool. We will talk in class about it. Write down questions right now. Not later, now. Hurry up, do it. I don't have all day. Okay, good. Now you've got it written down. Ask me those questions tomorrow in class. All right, have a great day.